Hello everyone, my name is Marc Nunez and I'm a fourth year undergraduate student from the University of Toronto's Pharmacology and Toxicology Department. In this video, I would like to present my research project, which focused on using the ICM protein modeling and computational software to analyze and identify potential inhibitory ligands for an endosomal protease called Cathepsin L. Since the first several COVID-19 cases were reported on December 2019, approximately 135 million cases of COVID-19 have now been recorded worldwide. While the development and deployment of COVID-19 vaccines represents an important milestone in fighting the COVID-19 pandemic, as of April 6, 2021, increasing COVID-19 cases have been reported in Southeast Asia and several regions of North America and Europe. With reported COVID-19 deaths continuing to increase globally, research into additional drugs against COVID-19 continues to be an important field of research. In identifying potential therapeutic interventions to help treat COVID-19, examining the replication cycle of SARS-CoV-2, the virus that causes the COVID-19 disease, can help identify important processes that may be targetable by drugs. It has been elucidated that the spike proteins present on the surface of SARS-CoV-2 virions can bind to ACE2 receptors present on host cell surfaces, such as the type 2 alveolar epithelial cells in the lungs. Primarily, the binding of the spike protein to ACE2 receptors leads to the processing of this viral protein and subsequently leads to the release of the viral RNA into the host cell. In addition to this cell entry mechanism, however, the whole SARS-CoV-2 virion can also be endocytosed into host cells. Similar to previous SARS-CoV viruses, cysteine cathepsin L proteases found in endosomes can also cleave the viral spike proteins of SARS-CoV-2. This process then also reveals the hydrophobic fusion peptide that allows for viral entry and infection of the host cells. Therefore, by inhibiting the activity of cathepsin L, this could present a novel therapeutic intervention that could help reduce SARS-CoV-2 infection, especially if it is paired with inhibitory drugs for other key proteases such as TMPR, SS2, which are also important for SARS-CoV-2 viral cell entry mechanisms. Therefore, for my research project, in silico techniques and approaches involving the ICM computational software were used to characterize and identify novel cathepsin L ligands and inhibitors. Structure-based approaches will be utilized to examine the active site of cathepsin L and to identify important ligand protease binding interactions. This information would thus be used to potentially identify ligands that can undergo high affinity binding with cathepsin L. In addition, ligand binding approaches will be used where chemical structures and properties of known cathepsin L inhibitors will act as templates in order to search for novel cathepsin L ligands. To start off, the active site of cathepsin L, which is shown here in the ICM slide, was first examined and was found to be consistent with findings from other research studies. As shown in this ICM slide, the active site, which is represented by the green area, takes on the form of a shallow and elongated binding cleft that runs through the middle of this protease. As illustrated by the ICM software, there are two major regions of the active site that can be identified. The blue region represents the S1, S2, and S3 binding sites, while the red region represents the S' active site regions. Based off available crystal structures obtained from the Protein Data Bank, or PDB, depicting ligand binding to cathepsin L as shown in this slide, it is apparent that the S1 to S3 sites are often critical in ligand binding and have distinct characteristics. On the other hand, the S2 site forms a small pocket lined with hydrophobic residues such as leucine 69 and methionine 70, which influences the binding affinity and selectivity of cathepsin L ligands. Lastly, ligand can interact with the S3 site to improve their binding to cathepsin L. For example, favorable pi stacking interactions to the glycine 67 to glycine 68 planar peptide backbone may be utilized. Although the S' regions do not typically interact with the smaller compound ligands, other ligands can also occupy these S' sites if they are large enough, allowing for cathepsin L to accommodate the binding of larger ligands such as polypeptide chains. After examining the active site of cathepsin L, virtual screens were then carried out to examine key interactions formed between ligands and the cathepsin L active site residues. Specifically, docking simulations were performed by ICM to model how ligands and compounds could bind in an optimal and energetically favorable manner to a protein of interest. In the ICM video, the pink structures represent the active site residues of cathepsin L, while the blue structure shows how an inhibitor is known to bind to cathepsin L according to available crystal structure data from the PDB. ICM will then flexibly dock the ligand, as shown here by the yellow structure, onto the specified active site residues and calculates the docking score for the modeled binding poses. These docking scores assess the binding pose under numerous criteria, such as changes in binding free energy and ligand-protein hydrogen bond formation. In this video, ICM was able to predict and recreate the known binding pose of the inhibitor, as the binding pose of the yellow structure and the blue structure are quite similar. Overall, lower and more negative docking scores represents better ligand binding to the protein of interest. In order to examine important binding interactions between ligands and cathepsin L, a compound library containing 2042 small diverse chemical fragments were docked onto cathepsin L's active site. After the completion of the virtual screen, the distribution of the docking scores obtained from this fragment-based compound library is depicted in the graph. The majority, or 56.6% of the chemical fragments in the library had docking scores in the range of -20 to -10, based on the virtual screening results. In addition, the mean docking score from this screen was approximately -12. Overall, these results do not represent favorable ligand binding, as other proteins with deeper active site binding pockets can have docking scores in the range of -30 to -50. Despite the distribution of the docking scores, there were 148 ligands, or approximately 7% of the fragment library, that had docking scores of -20 or lower. The binding poses of these compound fragments were subsequently analyzed to determine if there were certain interactions with the active site that were commonly being formed, which could have resulted in these ligands' higher binding scores. As shown in the ICM slide, there were 9 residues with which the top 200 best scoring fragments were frequently forming hydrogen bond interactions. When expanded to the entire fragment library, 3 residues in particular were identified as interaction hotspots, as the aspartic acid 71, methionine 70, and glycine 68 residues formed hydrogen bonds with at least 20% of all the docked fragment compounds. Following the fragment-based docking results, an additional virtual screen involving a chemical library containing 11,942 drug-like compounds was also performed. As the 3 interaction hotspot residues frequently form hydrogen bonds with both the top-scoring chemical fragments as well as the entire fragment library, the aspartic acid 71, methionine 70, and glycine 68 residues were added as docking restraints for this docking simulation. As a result, ICM prioritized binding poses that allowed ligands to form hydrogen bonds with these 3 interaction hotspot residues. The distribution of the docking scores from the drug-like library virtual screen were also plotted in the graph seen in this slide. The mean docking score from the screen was at around -9.4, and thus it yielded poorer results than the previous fragment-based virtual screen, which had a mean docking score of around -12. In addition, an increased proportion of ligands had docking scores of around 10 to -10, compared to the previous fragment-based library screen. This may be due to the prioritization of forming highly strained, non-optimal ligand binding poses in this docking simulation to satisfy the implemented docking restraints. Overall, this structure-based approach, where ligands were forced to form hydrogen bonds with identified interaction hotspot residues, was not successful in identifying ligands that can bind with high affinity to cathepsin L. For the following screens, ligand-based approaches were utilized to find novel compounds that can potentially become cathepsin L inhibitors. For example, RIDE searches were performed where ICM screened an enamine database containing approximately 2 million chemicals to identify compounds that had similar chemical properties as known cathepsin L ligands. In the image below, ICM has identified the distinct chemical properties of the depicted cathepsin L inhibitor, such as regions containing hydrogen bond donors, which are represented by the blue regions of the image, as well as the lipophilic regions of the chemical, which is also demonstrated or illustrated by the yellow regions of the chemical structure. Additionally, bioisosteres were generated using known cathepsin L inhibitors from the PDB as the structural templates. In this process, ICM substituted specified portions of the template structure with different atoms and functional groups to produce new and varied compounds. In the ICM slide, the yellow compound is a known cathepsin L inhibitor, which was used as a template to produce bioisosteres. As we can see in the following slides, ICM produced bioisosteres, which are shown in the non-yellow colors, and if we look at the rest of the structure of the known cathepsin L inhibitor, we can see that the bioisosteres share the same chemical structure except for the para-methyl substituent that is present on the template's phenyl ring originally. And as we can see here with the other two bioisosteres, the same is found as the chemical structure is the same except for this substituent as well as for this final and third bioisostere. Lastly, ICM was used to build a 3D QSAR model which could predict the inhibitory activity of a given compound after comparing its chemical properties to those of a template. 3 cathepsin L inhibitors from the PDB were used as the model templates as seen in the slide here. Similar to what was seen with the RIDE searches, ICM was able to visualize and merge the chemical properties of the 3 model templates into one figure. Similarly, a Campbell compound library containing 1,136 compounds with reported inhibitory activity or p-activity data was used to build the predictive model. The graph in the slide shows that in comparison to the known p-activity data for 500 Campbell ligands included in the data set used for testing, the 3D model predictions were only slightly effective in predicting ligands' known p-activity values. As the predicted p-activity values often varied and did not match the known p-activity values for the Campbell ligands, this resulted in a coefficient of determination, or R-squared value, of only 0.33 for this 3D QSAR model. After compiling RIDE search hits and bioisosteres, the resulting library containing 7,349 unique compounds were docked onto cathepsin L. Unlike the previous virtual screen of the drug-like compound library, this docking simulation did not include any docking restraints. Overall, this virtual screen, incorporating ligand-based approaches, had a mean docking score of -15.2, which was the lowest and best docking score obtained amongst the performed virtual screens so far. In addition, the percentage of the library with docking scores ranging from -30 to -20 was 3 folds higher than the previous fragment-based and drug-like library virtual screens. Based off these results, incorporating ligand-based approaches were more successful in finding ligands that can more favorably bind to cathepsin L's active site compared to the previous structure-based screens. In addition to assessing the bioisosteres and RIDE hits docking scores to find favorable cathepsin L ligands, the 3D QSAR model was also applied to this compound library to predict the inhibitory activity of each ligand. Compounds with high p-activity values correspond to stronger inhibitor affinity and activity to proteins and enzymes, and should thus also have low docking scores. Subsequently, the graph in this slide shows how the predicted p-activity values compare against the docking scores of the ligands from the virtual screen. It is apparent that there is only a weak association between the predicted p-activity values and the docking scores, resulting in a low r-squared value of only 0.07 for this graph. Despite the poor association, there were 34 ligands that had p-activity values greater than 6, which is equivalent to an inhibitory constant or IC50 of about 1 micromolar, and also had ICM docking scores lower than -30, which is shown here in the highlighted portion of the graph. Notably, these 34 ligands are specifically the ligands that have the best docking scores from the ride and bio-isosteer virtual screen. Therefore, both the virtual screen and the 3D QSAR model were able to isolate the same set of compounds, which they predicted will provide high affinity or effective inhibition of cathepsin L. When analyzing the interactions that these top scoring ligands form with the cathepsin L active site, it was noted that they also form hydrogen bonds with the interaction hotspot residues identified from the previous fragment-based library virtual screen. Specifically, the top scoring ligands also form hydrogen bonds with aspartic acid 71, methionine 70, and glycine 68 residues. As shown in the graph, however, there were additional active site residues in the S1 and S' binding site regions that underwent hydrogen bonding with these top scoring ligands as well. As seen in the slide, these residues were specifically aspartic acid 162, glutamine 19, and the tryptophan 189 residues of the cathepsin L active site. Based off the virtual screen results and examination of their binding to cathepsin L, several properties and interactions of these top scoring ligands were incorporated into the design of a new cathepsin L inhibitor. To start, the binding pose of the ligand with the best docking score from the ride and bio-isosteer library virtual screen, which is shown here in this slide, was used to serve as the base template for the new ligand. As we see in the next slide, it already forms hydrogen bonds with the glutamine 19 residue and also forms favorable pi stacking interactions with the histidine 163 residue. This is a common interactions found amongst the top scoring ligands that have chemical structures in this region of the active site of cathepsin L. From other ligands, the heterocyclic and aromatic oxidiazole substituent was included to project further into the S2 pocket and form hydrogen bonds with the aspartic acid 71 and methionine 70 residues in this region. In addition, a large addition to the chemical structure was added to the novel inhibitor, as this allows it to occupy the S3 binding site and form hydrogen bonds with the glycine 68 interaction hotspot residue as seen in this slide over here. By combining all these key interactions from other ligands, which are shown in the purple regions of this molecular compound, a novel compound was formed which had a docking score of -43.7 when screened against cathepsin L. Compared to the lowest docking scores from previous screens, which range from -32.5 to -36.5, this new ligand has the best docking score result and would likely bind with high affinity to cathepsin L. This could allow for competitive inhibition of cathepsin L, as the ligand occupies the catalytic S1 binding pocket of the active site, which is represented here in the ICM slide by the red area of the receptor mesh, when this novel inhibitor is bound to cathepsin L. As an alternate view, if we display the space that the novel inhibitor takes up in the active site of cathepsin L when it is bound to the protease, we can see that it firmly fits into the elongated binding cleft active site of cathepsin L, And you can see here that even in this view, the catalytic cysteine residue of the protease is covered up by the binding of this novel inhibitor. In addition, according to the 3D QSAR model, this new ligand also has a predicted P activity value of about 8.5. The ligand could thus prevent the proteolytic degradation and cleavage of other ligands, such as endogenous proteins or exogenous viral proteins as well. Unlike other known cathepsin L inhibitors, the inhibitory activity of this ligand will likely be reversible as it does not covalently bind to the catalytic cysteine residue of the protease. Overall, these results therefore provides additional evidence and support that this ligand could be an effective inhibitor against cathepsin L. Based on my research findings, incorporating both structure-based and receptor-based in silico approaches and techniques allowed for the identification of key interactions important for the high affinity binding of ligands to cathepsin L. These findings ultimately allowed for the generation of a novel inhibitory ligand against cathepsin L, which can bind favorably to its shallow active site and has a high predicted P activity value according to the formulated 3D QSAR model in this study. Ultimately, this novel ligand could be a promising candidate for further lead optimization and drug development, which could potentially help treat patients with COVID-19 or other diseases related to cathepsin L in the future. And overall, these are my references. And I would like to thank you for listening. And I would like to especially thank Dr. Matthew Shapira for supervising my research project course and for always providing his help and insights while I was conducting my research. Thank you.